A new DNA Nano engine has been created by a team of international researchers. A new kind of nano engine made of DNA has been created by a team of scientists from around the world. This engine operates through a clever mechanism and is capable of pulsing movements. The scientists are currently exploring the possibility of adding a coupling to the engine and integrating it as a drive in complex nanomachines. Their findings were recently published in the journal Nature Nanotechnology. The project in question involved the cooperative efforts of 3 distinguished scholars. Peter Sulk, an assistant professor at Arizona State University's School of Molecular Sciences, collaborated with Professor Famulok, who served as the project lead from the University of Bonn in Germany, and Professor Walter from the University of Michigan. Using computer modeling tools, Sulk and his team have gained valuable insights into the workings and design of the leaf spring nano engine. The structure of this engine is composed of approximately 14,000 nucleotides, which are the fundamental building blocks of DNA and serve as the basic structural units. According to Sulk, the computer model OxDNA is essential for designing and simulating motion in large nanostructures. Without it, such feats would be unattainable. Recently, our research group was able to successfully engineer a DNA nanotechnology motor that is powered by chemical reactions. This is a groundbreaking achievement, as it is the first time this has been accomplished. We are thrilled that our research methods can aid in the study of this extraordinary nanomachine and are eager to construct even more intricate nanodevices in the future. This innovative engine design bears resemblance to a strength training tool that enhances hand grip when used consistently. Nonetheless, the engine is astoundingly minuscule, measuring approximately 1/1,000,000th of its counterpart size. The engine comprises 2 handles that are joined by a spring, forming a V-shaped configuration. The act of squeezing the handles in a hand grip strength trainer involves applying force against the spring's resistance. Upon releasing the grip, the spring proceeds to push the handles back to their initial position. Professor Michael Famulok from the Life and Medical Sciences Limes Institute at the University of Bonn explains that their motor operates under a similar principle. However, instead of pressing the handles together, they are pulled towards each other. A mechanism that is fundamental to the existence of all life on Earth has been repurposed by researchers. Each and every cell within an organism has a library of sorts, storing the essential blueprints for all the various proteins that the cell requires in order to carry out its designated functions. When a cell necessitates the production of a particular protein, it orders a copy of the corresponding blueprint, which is then transcribed by RNA polymerases, the enzymes responsible for this process. The original design is made up of extended DNA strands. RNA polymerases traverse these strands and replicate the saved data, letter by letter. Famulok, a member of the interdisciplinary research departments Matter and Life and AMP Health at the University of Bonn, elucidated that they affixed an RNA polymerase to one of the handles on their nanomachine. In close proximity to this, they stretched a DNA strand between the 2 handles, and the polymerase attached itself to this strand in order to replicate it. By pulling itself along the strand, the non-transcribed portion becomes progressively smaller. Consequently, this pulls the second handle incrementally closer to the first, while also compressing the spring. The DNA strand contains a specific sequence of letters prior to its termination point within the handles. This sequence, also referred to as the termination sequence, serves as an indicator to the polymerase that it should release the DNA. Consequently, the spring is allowed to relax and the handles separate, bringing the start sequence in close proximity to the polymerase, thereby enabling the molecular copier to initiate a new transcription process. This cycle continues in a repeating pattern, resulting in a pulsating action by the nanomotor. Matthias Centola, from the research group led by Professor Famulok, who conducted a significant portion of the experiments, explains this process. Similar to other types of motors, this motor requires energy, which is provided by the nucleotides produced by the polymerase from the alphabet soup. Each nucleotide has a small tail that contains 3 phosphate groups, known as a triphosphate. The polymerase must remove 2 of these phosphate groups to attach a new letter to an existing sentence. Famulok explains that nucleotide triphosphates serve as the fuel for the motor, releasing energy that is utilized for linking letters together. The motor is only capable of running when an adequate amount of these triphosphates are accessible. Through their research, the scientists were able to illustrate how the motor could be seamlessly integrated with various other structures. This opens up the possibility for the motor to move across surfaces, much like the manner in which an inchworm crawls along a branch. Famulok elaborates on their future plans, stating that they intend to develop a clutch mechanism that will enable them to control the use of the motor's power, allowing it to idle when not in use. As a long-term goal, the motor could potentially serve as the central component of a sophisticated nanomachine. However, there is still a great deal of work that needs to be done before this vision can be realized. The laboratory of Salk is characterized by its interdisciplinary nature, utilizing statistical physics and computational modeling techniques to address a wide range of problems spanning the fields of nanotechnology, chemistry, and biology. Specifically, the research group is dedicated to constructing novel multi-scale models, which are used to examine the interactions that occur between biomolecules, with a particular emphasis on the design and simulation of DNA and RNA nanostructures and devices. The molecular sciences, much like the everyday use of complex machines such as cars, planes, and electronic chips, necessitate the utilization of advanced computer-aided design tools. These tools are crucial in guaranteeing that the molecular components perform their intended functions with precision and accuracy. According to the director of the School of Molecular Sciences, Professor Tijana Raj, Peter Salk and his team are conducting highly innovative research in molecular science. Their approach involves utilizing computational chemistry and physics to investigate the properties of DNA and RNA molecules, both in the context of biology and nanotechnology. Professor Salk serves as an excellent example of the exceptional accomplishments of the younger faculty members at the School of Molecular Sciences. DNA and RNA are fundamental molecules that play numerous roles in living cells, such as storing and transmitting information. These molecules also have potential applications in the field of nanotechnology, where engineered DNA and RNA strands are used to construct nanoscale devices and structures. As Salk describes, the process is similar to constructing with Lego blocks, although each block is only a few nanometers in size, and the blocks are placed in a box and shaken until the desired structure emerges. The procedure of constructing shapes at the nanoscale level is referred to as self-assembly. Salk and his associates utilize computational modeling and design software to create the particular building blocks that have a high likelihood of assembling into the desired shape with precision at the nanoscale level. According to Salk, the potential uses of this area of study encompass diagnostics, therapeutics, and the creation of novel materials. His laboratory has created the necessary software to design these building blocks, and they collaborate intimately with experimental teams at ASU, as well as other academic institutions throughout the United States and Europe. The funding for this research project was provided by the ERC grant with the identification number 1010400035. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.